Hi, welcome to my presentation. My name is Amanda Piapani, and I'll be talking about my research analyzing the nervous system of leeches. I worked with the Wagenaar Lab at Caltech, and my mentors were Dr. Daniel Wagenaar and Dr. Pega Kasarayan-Fard. Within the scope of the Wagenaar Lab, I was interested in how nervous system structure supports behavior. This covers neurite growth processes, synaptic clustering and generation behaviors, as well as modeling neuronal activity patterns as a function of synaptic input patterns. The nervous system is composed of neurons that transmit information throughout the body. In order to do that, neurons work together in neural circuits that follow very specific pathways. Neurons generally have a cell body, axon, and multiple dendrites. For leeches, there isn't a structural distinction between axons and dendrites, so we'll be referring to those as projections as neurites. The structure of the neuron that we're really interested in are the synapses, which are sites at which the neurons relay information and are generally located at the end of an axon for a presynaptic neuron and at the dendrites and cell body of the postsynaptic neuron. So here we have the cell body, and this is the neurite along with its branches. So the terminology we use to understand neurites is based off of graph theory. First, we start off our tree at the cell body, and we have neurites that grow from that cell body. A node is a branching point which connects adjacent segments, and a segment contains a node at its beginning and end. Parent segments are connected to child segments by their end node, and child segments are connected to their parent segments by their beginning node. So for some segments, they're only parent segments, some are only child, and some segments can be parent and child segments. Intermediate segments are any segments that have at least one child, and terminal segments are segments without any children. So as you can see here, in red we have the terminal segments, and in blue we have the intermediate segments in this example. So what does the field currently know about neurite morphology and how it affects output? So unlike in the field like physics, we don't have a strict formula for where and why synapses form at the locations they form at. We currently know that neural circuits process information differently by the types of neuron and how their synapses are distributed. For example, studies over the past decade have shown that certain neuron models match biology better when synapses are randomly allocated than clustered and vice versa. In the rat, there is evidence that shows that hippocampal cells work better with randomly allocated synapses, and pyramidal neurons work better with clustered synapses. Today, we want to focus on our work analyzing neurite growth processes. Growth processes refer to how segments develop from adolescence. We understand the growth process through analyzing the distributions of terminal and intermediate segments. When we get a better understanding, we can create a model of how the segments develop through stimulating probabilities of features such as growth, branching, stopping, and retracting. So we're really interested in studying the leech, specifically the medicinal leech. We study them because as invertebrates, they have a relatively accessible nervous system. They also have relatively large neurons that yield strong output signals, and they express identifiable behaviors, which is really important because our trials are divided into different behaviors. We focus on the leech's DE3 motor neuron and three key behaviors: swimming, crawling, and an escape reflex we call local bending. All of these behaviors share a common output in the DE3 motor neuron, and each of the behavior's neural circuits include DE3. So our research is primarily focused around DE3R and its presynaptic partners. So here's a quick video showing the structure of DE3R. So here in white, each of the nodes represent a node, and each of the lines represent a segment, and every color represents a different presynaptic partner. So in order to pursue our research goals, we needed two key things about DE3. First, we needed to be able to record DE3R's activity during each of the three behaviors. And second, we of course needed to be able to record DE3 structure. So how did we do this? First, we used voltage-sensitive dyes to record neuronal activity. If you're familiar with action potentials, VSDs allow us to record the subthreshold neuronal activities instead of just the spiking. This activity is what we refer to when we meant that the leech neurons yield strong output signals. By using VSDs, we were able to show that synapses work in clusters for different behaviors, which we'll show on slide 18. So here we have two examples of VSD traces of swimming on the left and crawling on the right. These are measured through change in fluorescence over time, and as you can see, DE3R has a different trace for different behaviors, which tell us that the cell is operating differently during swimming versus crawling, and potentially other behaviors. Second, we use serial block phase electron microscopy to anatomically image the ganglion, which is just a group of neuron cell bodies that DE3R resides in. This technique was really important because it allowed us to also get the location of the synapses. Here we can see the raw biological data that SBEM was able to measure. And if you're interested in seeing this further, you can head over to leachem.caltech.edu to check it out. So here in green are the outline segments that were traced, and this process was so painstakingly difficult that it took 2 years to complete. If you compare it to the previous slide, it's not immediately obvious where these should reside. I'll introduce the most important visualizations made during my research. So first we have the segment distributions by type, with the intermediate segments on the left and the terminal segments on the right. So here we have it, frequency on the Y and length in microns on the x-axis. And we found that the log normal distribution fits both the intermediate and the terminal segments the best based on sum squared error. And this is really essential to understand the growth process with, as we mentioned earlier. And we found that terminal segments tend to be shorter. So there could be a multitude of explanations for this. When we refer it to in terms of the probabilities we talked about earlier, we could have the probability of retraction being higher, or we could have the probability of branching being higher. Of course, these are just two explanations for what could be happening. So second, we have the terminal segment length and edge distribution. And on the left, we have terminal segments with only one or more synapses. And on the right, we have terminal segments with 0 synapses. So we're really interested in seeing if synapses had a role in stabilizing the growth process. However, we didn't end up finding a correlation between synapses and if the growth process was stabilized. However, something else was really made this graph important because it helped us find a measurement bias in our data in the first iteration of this graph. So if you see in the bottom left of both graphs, you can see where segments with one edge of this length, which is actually 1 micron, is cut off in both corners. And this is actually intentional because we found that by graphing this, when the first iteration, we found that we had an overrepresentation of terminal segments with 1 synapse of edge length 1, which is kind of a mouthful. But this was because we had entities in the database that appeared to be terminal segments, but were simply just synapses sitting on the wall of a parent neurite. After doing some data cleaning, we found change results in the previous distribution. However, it remained the same. Here is just a preview of the 3D model of DE3R and synaptic clusters for the swimming experiment. And when compared to other behaviors, the clustering actually looks different because the neuron is operating differently. Lastly, one of the projects that we worked on that I won't have time to really go into today was our test to see if we could predict DE3R output based on presynaptic inputs. I've only been able to talk about growth processes in depth, but I look forward to talking about other subjects in the future. This research was made possible by the Caltech SURF program at the Wagenaar Lab, and I would like to thank my mentors again, Dr. Daniel Wagenaar and Dr. Pega Kasarayan-Fart, for the amazing opportunity to work with them.